How are you? I know, how are you? Yeah? Okay. He is. He's really fun. Good evening, everyone. My name is Emilia Huerta Sanchez, and I am one of the co-organizers. Today, it is my pleasure to introduce Magnus Nordborg. Magnus is the director of the Gregor Mendel Institute in Vienna. He received his PhD from Stanford University. Then he did a postdoc at the University of Chicago, moved to Sweden, but after 3 years moved back to the US and took a faculty position at USC, and then moved to Vienna in 2009. Magnus has made several significant contributions to our understanding of how evolution works. He's used population genetic theory, has developed statistical and computational methods, and applied them to genomics data to address questions such as understanding the link between genotype and phenotype, the genetic basis of adaptive traits, the genetics of species differences, and today he will tell us about his work on the genetics of epigenetics. Thank you, Magnus.
Thank you. Thanks so much. I hope everyone can hear me here. I mean, the masky thing and everything. It's absolutely great to be back in Asilomar. This is really one of the best places to have meetings, right? It's so beautiful. It's great to be back in, in-person meetings as well. Didn't miss the jet lag, so I hope I don't fall asleep during my own talk. If I do, or just have a senior moment, please somebody wake me up. Okay, so right, I'm going to talk about something very different, but first, let me see here. Wait. Now what happened? Right. So yeah, this is where I work. Just a quick advertising slide. It's a nice place to work. This is the city in which I live. It's also a nice city. And then given this audience, I thought I should say the proudest thing I am about the Institute. We have the world's best logo, arguably the only logo that makes any sense. I hope everyone sees this. So I'm going to talk about Arabidopsis, which, it's the premier plant model. And even though it has sort of been quite defunded much in the US, it's quite interesting if you work in plant genomics now. Almost all the annotation that exists for any plant anywhere really comes from Arabidopsis. It just gets lifted umpteen times from the organism where it was originally developed. And so I started getting into working with this several decades ago, and my interest was always in natural variation. And so this plant grows out in the wild as well. And these are typical habitats. And this is what it looks like when it grows outside. In biology, it's a typical weed, not a dangerous weed, right? It doesn't actually compete with any crops. In fact, it doesn't compete with anything. This is the typical plant that really is the world's worst competitor, really. It can only grow where there's some kind of disturbance. Animals, natural, on mountain slopes like you have on the right here, or animals kicking up things, or near rocks, or out on beaches where wind is constantly blowing things around. Most patches where you find Arabidopsis, where humans have disturbed it, they will be overgrown with grass in just a few years, and the whole population is gone again. And so the thing that got me and several other people interested in this early on is that, unlike, say, Drosophila, the fact that you have these natural inbred lines, right, is that you can collect plants from all over the world, adapted to their local habitats, bring them in and work on them, and have a germplasm that everyone can share. And it was pretty clear early on that this would be an ideal thing for doing things like genome-wide association, right? Because you just need to sequence these lines once and so on. And so and there's a lot of people to mention here, spent a lot of time going through effectively the same steps that happen in the human genetics community, trying to figure out what the population structure was like, how to deal with that, what was the extent of linkage to equilibrium, figured out that you can actually do genome-wide association. We developed SNP chips. By the time we had the SNP chips, they were obsolete because then you could sequence things already. So we've done next generation sequencing. There's a large set of over 1,000 lines available, for which SNPs are called from SNP genotyping with short reads, right? And the next thing we're doing now is trying to sequence these genomes completely. So I guess we're up to about 200 genomes completely assembled now. I'm not going to talk anything about that today. But so, yeah, there's a bunch of people here involved in creating the 1001 Genomes project. Basically, it's actually more than 1001 genomes available, but the key here is that there is a stock center. You can order the seeds, you can keep the seeds in the tube for years, just grow them whatever you want. And our goal is to create as complete genomes for all of these as possible, and then pile on as much phenotype data as we can. Of course, we cannot at all keep up with, say, the UK Biobank in the sort of scale of anything, but you can, but nonetheless, I would say this, that genome-wide association studies, they work as they were originally advertised in human genetics in Arabidopsis, in the sense that when you map things, and the Manhattan plots here show GWAS flowering time, and you find big genes that have big effect and are pretty often easily interpretable. They have known pathways, and you can do experiments on them and so on. So that's sort of the background. But what I'm going to talk about today is epigenetics. So what is epigenetics? Well, it's one of those horrible terms that gets flung around a lot. Typically, we're referring to some kind of inherited cell memory, right? In particular, across generations. And I'm going to talk exclusively about DNA cytosine methylation, and which has become this thing in parts of evolutionary biology. People think about it as a fifth base that sort of does mysterious gene regulatory things and get transmitted around. And I go to meetings sometimes where you have people who talk about this from an evolutionary point of view, and they try to bring them together with molecular biologists who work on this. And there's a complete lack of communication, because they use the same word to describe something completely different, seemingly. And so, but somehow the notion here is that environment can change DNA methylation, and that this then sort of can be passed into the next generation in some kind of Lysenkoism, Lamarckism, right? And I put up the picture of Lysenko here. I mean, this is sort of I mean, he looks nice, doesn't he? Hollywood will have to find a Swedish actor to play him in the next movie. Well, underneath it, of course, is Nikolai Vavilov, who truly was a great geneticist, did expeditions all over the world, did enormous work on domestication of many species, and was eventually, partly thanks to the machinations of Lysenko, thrown in jail, and he died in World War II. Right, okay. But so the whole thing about, okay, so where do these notions come from? So just a few factors here now, and I apologize. There are almost always someone in the audience who knows more about this than I do, but okay, DNA methylation. So in humans, right? So DNA methylation is an important biomarker for cancer, for smoking. You can tell a lot of things just from checking methylation status of blood, for example. And almost in most organisms, methylation of promoters is related to repression of expression, right? And it's also in most organisms associated with silencing of transposons. I put these words in italics because one of the things about methylation is that it doesn't really by itself, so vast oversimplifications here, but doesn't by itself do very much, right? What it seems to be is a tag on DNA that helps attract histones that are modified in the right way. And those are the things that actually do things, right? And this is kind of clear because you find DNA methylation in Arabidopsis and in humans on heterochromatin, right? But of course, methylation is not universal. Flies, for example, don't have it, but they do have heterochromatin. So clearly it's not fundamental for this purpose, right? It's like an extra layer of chrome on top of this. Other important things to note is in vertebrates, methylation is erased almost exclusively during meiosis, right? So in terms of being an epigenetic memory, it doesn't quite work. However, in plants, it is absolutely not erased. And so this tells you right away that the idea that methylation plays a role in development can't quite be right for both of these, right? Because if you want to have something involved in development, you kind of want to erase it, right? And that seems to happen in animals. I mean, if you knock out methylation, as in the mouse embryos on the left, animals die. But Arabidopsis, you can knock out methylation and you still get a plant, right? So that kind of is a pretty strong proof that it's not a very important developmental regulator. You don't get a happy plant, and after a few generations, that's what you see here, they get progressively sicker and they don't live long. And in many plants, they die immediately, right? And that's probably related to how many transposons they have. Okay, furthermore, in plants, you have methylation in 3 different sequence contexts. So in animals, there is only CG methylation for the most part, or mainly, I should say. So CG methylation is symmetric DNA, right? So when the DNA gets replicated, both strands are hemimethylated, and then there's an enzyme that comes back and puts it back on again. And this enzyme is the same in both plants and animals, and it works the same way. But plants also have CHG-methylation, so which is also symmetric, right? So H here stands for anything but a G. And this stuff can also be passed on. There's a feedback loop involving chromatin modification, so this can be fixed back on again. But there's also any C can be methylated, because CHH state here, right? And that, of course, would get lost automatically during cell division unless it's put on actively. And you can see this if you look at the graph on the right. Let's see, I had problems with this before. Yeah, so now the mouse is not, whoops, knew that was going to happen. All right, never mind. I'm going to use this anyway. That doesn't work either. Okay, I'll just describe the figure. Okay, so on the figure on the right, you see the distribution of methylation. So, of course, in any given cell, right, the C is either methylated or it isn't, right? But when we assay this, typically we grind up all plants with millions and millions of cells, right? So you can look at the fraction of methylation at each site. And the red line looks at CG sites. And you can see that most sites are either not methylated or almost completely methylated, right? That's what you'd expect to see for something that's been inherited, right? Just like a SNP. You either have this allele or you have that allele, and that's true for all cells in your body. But the other types of methylation don't show this pattern at all, right? You have often quite low levels of methylation and intermediate. So this is hard to reconcile with this having been passed on as an epigenetic mark. Okay. You also find in Arabidopsis, we can think about methylation in two quite different configurations. There is something that's associated with heterochromatin, so transposon-like methylation. And this in the regions, so this is characteristic for transposons and other repetitive regions. You find heavy methylation in all contexts. And yeah, so heterochromatin, and this is generally associated with repression. And the pathways, there are known pathways that establish this, the RNA-directed DNA methylation pathway. So small RNAs target enzymes 2 pieces of DNA and it gets silenced, right? CMT2 pathway directly puts on CHH-methylation in heterochromatic regions. And this stuff can also be maintained, and this involves then the histones being modified, and there are many different ways. It's quite complicated. In addition, we also find gene body methylation, which at least superficially looks very similar to what we find in mammals. So this is especially on highly expressed housekeeping genes. You find sparse CG sites inside these. And that tends to be positively correlated with expression. You know, these are highly expressed genes, so in that sense. And why it's there, how it's established is debated, and nobody really knows. Okay, so how did I get into this? Well, if you go back to the slide with Lysenko, so many years ago, we were going to do a study of how genotype translates into phenotype. We did transcriptome profiling, and I think it was Richard Clark who said, let's try to do bisulfite sequencing on all these lines. So this was a relatively small sample, but we did. And we just wanted to ask the question, how can you combine these different layers of information? So we asked the basic question here, does methylation affect gene expression in a quantitative sense in plants? So small sample, the Swedish Arabidopsis, we had genomes, we had bisulfite sequencing, so methylome, right? And we had transcriptome. And so then you do things like this. So here is a GWAS on 20,000 transcriptome profiles, and on the y-axis there are 5 chromosomes. You see 5 different axes there, and on the x-axis are the SNP positions. And what you're looking at for each, so you have 20,000 quantitative traits going across horizontally, and the red dots are the significant peaks, right? So you see a long band along the diagonal, that's all the cis regulatory variation, right? So these are SNPs associated with expression variation, so eQTLs directly, right? If there were any major transacting regulators, they would be vertical lines going down like this. We see bands going this way across, and those are artifacts. That basically means that expression for this particular gene has such a weird distribution that the GWAS throws out false positives all over the place. Okay, there are lots of dots. And then we can play the same game with methylation variants instead of SNPs. So now axes are the same, except we're looking at methylation variants, and the pattern looks similar. You have cis regulatory stuff, you have trans regulatory stuff. There are many fewer. That's one point. The other interesting point, and this was completely unexpected, here we actually find a clear trans regulatory thing, and this turns out to be one of the Argonauts, which is involved with processing small RNA. So this is pretty cool, right? So this is a bona fide epi allele. Methylation on AGO4 is associated with expression of a whole bunch of genes, and there were no SNPs capturing this. So this is a real epi allele. But there are not very many of those. And if you try to fit all of this together, so here is you take all the genes in the genome, and you fit the model where you partition the variants into local methylation status, local SNPs, and genetic background SNPs, right? Because there's a lot of trans regulation of expression, of course. We bin them here by how well the model fit, how much of the variation you explain. And on the top, you can see a histogram that for most of the time we're explaining like maybe 20% of the variation here. But you see the green bar is overwhelming. Most of the regulation is trans-regulatory stuff for most of the genes. But the main thing is that the methylation explains extremely little here, right? So from a quantitative point of view, it doesn't explain very much. So, okay, so that was in a way kind of disappointing because we thought that we would have some cool information. There are obviously many cool examples where methylation is doing something, right? And of course, I'm not saying that this is true under all circumstances. Maybe we get a different result if we shock the plant with, expose them to a pathogen or something like that. Then maybe methylation status would play a more important role. But at least under these conditions, it did not. However, we found something else that was completely unexpected. And that was for all types of methylation, we find extremely strong geographic patterns. We first saw this in the Swedish sample, where there was a difference between northern and southern Sweden in gene body methylation. But with the global sample, you start seeing these patterns everywhere, and they're correlated with every geographic variable, any climate variable you can imagine, and there's no real way to know whether any of that is significant. But patterns there are, and if you do sort of QST, FST-like things, it just doesn't, it does not look random at all. So this is interesting, right? Because, I find this immediately intellectually fascinating because, okay, what could cause this, right? Because what we're seeing here is that, so these are plants that are grown in growth chambers, and their methylation status tells you where they came from in some level, right? So somehow, either they have like an epigenetic memory of their origin, or there must be genes that are sort of putting this stuff on, right? Which also, and the genes then differ between their origin, right? So, you've got to figure out what are you actually looking at here, right? Are you looking at, should we view this stuff as some kind of phenotype or some kind of epigenotype, or what is it? Okay, and like I mentioned before, there are different types of methylation. If you look at the CG methylation, we know from numerous studies that it's pretty stably inherited. So the prevailing model in the literature, so there have been two types of studies here. One is looking at mutation accumulation lines. These are Ruth-Shaw's original lines growing plants over 30 generations, and just seeing how, they've been used to, they were originally meant for SNPs, right? But you can do the same thing then by sulfide sequencing, and you see that the methylation status remains constant. There are changes. There's a low rate of mutation. They estimate 10 to the -4 or something like that, right? So higher than SNPs, but still pretty stable. There's also been examples where you look at, for example, Arabidopsis invaded North America within the last 200 years, and methylation patterns seem to be sort of similar to where they were at the origin. There are also these famous epireals, a very clever construction. So one of the reasons we understand methylation so poorly is that we can't really manipulate it. Until recently, it hasn't been possible to manipulate it experimentally, right? You can knock it out in various ways, but that's like hitting the whole plant with a sledgehammer, so not great. These epireals are compromised. You knock out methylation, so you strip it, then you cross it back into a wild type, right? And get rid of the mutant allele. So then you have a mixture of unmethylated and methylated chromosomes. And these lead to quantitative variation. You can map things using just the epigenetics data. They're stable enough to do that. And they change over time, but again, are somewhat stably inherited. Okay, so that's CG methylation. However, if you look at the CHH methylation, it is clearly not a epigenotype like this. This is from our old study as well. We grew plants in 2 different temperatures. Each dot represents an inbred line, an accession. And so these are just reaction norms, right? So you see that the CG methylation kind of is the same regardless of which temperature we grew them in. But for CHH methylation, there's a direct response, right? So this responds directly to the environment. So this is clearly a phenotype. Okay, but of course it's complicated because these are not independent, right? Yes, CG methylation can keep on going by itself like this, but the pathways that put on CHH, which are temperature dependent, also put on CG methylation, right? And then when it's put on, it will probably stay there. And there are all kinds of interactions. So it's not that simple if you start thinking about how this might actually work. And then, of course, none of this would explain why you have these differences, right? So yeah, but I felt, and I'm not going to give you, I'm going to try to give some speculations about this, but I still don't know that, right? But before you can ask this question, you got to figure out mechanistically, what are you even looking at, right? I mean, this is sort of why I keep putting all these Mendel pictures up here. I mean, you have patterns of variation here, right? You have to have a mechanistic model for what's going on. So that's what we did. We started trying to dissect this, and we've been doing this for several years now, and I'm going to zoom through this. Okay, so genetics. So we did a GWAS, of course, because that's what we do. And if you look at, so the slide on the top two here, top two figures shows Manhattan plots for GWAS on CHH-methylation on transposable elements. And what they revealed, it was completely unexpected, a very, very striking architecture with small number of well-known genes explaining a lot of the variation. This is like 30% of the variation here, right? So major genes. This is like, you know, mapping skin color in humans or something like that, right? And these are two Argonauts. NRPE1 is a subunit of RNA polymerase 5, which is important for also for silencing and small RNA processing. CMT2 I already mentioned, and I call there are two types of TEs here, right? So of course you can knock out these genes, and you know which ones are targeted by that pathway or the other pathway. For gene body methylation, we don't see anything, and if you think about it, you shouldn't expect to see anything, because something that is stably inherited, whatever might have caused it, if there was a gene, it's not going to be linked to it anymore, right? So you're not going to expect to find anything there. Here's a slide I want to show. Okay, this one is a little bit complicated, but I show it anyway because I think it's so super cool, right? So this is like the strong claim of GWAS here, right? So what is this? On the, okay, the tree on the left here with the colors are transposon families. So 400 families of transposons. For each of those, we calculate the average methylation, and then we do a GWAS. And if you go up the, you know, the heat mappy thing is that, right? So the dark spots is where there's a significant hit. Then we cluster these, and you can think of these then, so people who work in human genetics, these are 400 phenotypes that are correlated. We cluster them by similarity in GWAS results. And you see that they fall into 4 different clusters here. There are 2 major ones. The second one from the top, there are a couple of bands, and if you go up on the top, we then calculate, you know, just add up the p-values with, you know, Fisher's method and all that stuff, and you recapture the results we had before. But you can see that there's one group here that is affected by CMD2. There is a big band underlying that gene. So, and then on the right, we line them up for each of these families, what happens if you knock out the relative genes, right? So the experimental results correspond to that. So, okay, so why do I think this is cool? Because this is kind of what we hoped that GWAS would be like, right? You can actually see the different pathways coming up here just by doing a GWAS result. So we know that the second cluster here are the RDDM targeted TEs, and the one below is the CMT2 targeted TEs. But that was 20 years of molecular biology to figure that out, right? And you could just see this directly from the GWAS result, right? So this is like, you know, really thinking about there being such major effects that you can view GWAS as a natural mutation screen where you actually learn the pathways and the architecture of the traits, right? And so that's like the strong version of how GWAS would work. And the hits are so strong that you can easily identify causal mutations in the various genes. I show NRP1 and CMT2 here. Okay, so that was CHH-methylation. Next, CHG. So for CHG, we didn't find anything when we did a GWAS, and we didn't quite know why. We didn't know what to do with it until Eriko Sasaki and my lab started poking around. And the first thing she noted is that CHG levels are strongly correlated with CHH. In this case, in mutants, right? So the plot on the left is the RDDM targeted transposons. The one on the right is the CMT2 targeted transposons. And the dots are different mutants from Steve Jacobson's lab. They've systematically knocked out all the known players in these various pathways. And what you see is that the phenotypes are massively correlated, right? less so for the CMT2 targeted ones. Okay, so that's interesting. But now what makes it really cool is if you look at the natural variation, now the lines are just random natural accessions and they follow exactly the same pattern, right? So clearly there is some mechanisms that forces you. We don't know exactly what it is that pushes you along this axis, but this axis is there for some fundamental reason, right? So these are correlated traits. Okay, so Eriko figured out that, okay, maybe if you need to analyze both these traits together. If you background statistical theory tells us that that would be the right thing to do, and it turns out to be the right thing to do. So the top plot shows GWAS when you of CHG-methylation, and you find nothing. And the bottom one is GWAS of CHG-methylation when you just add CHH-methylation as a cofactor in the model. And bingo, you get a repeat of what we have seen for CHH before. A small number of very obvious genes explaining a huge amount of the variation of this conditional phenotype, right? So I don't need to go into them. Some of these were previously known, but it's almost ridiculously clear. So there's a microRNA that regulates CMT3 that directly does CHG methylation. CMT2 has an they compete for the same targets. MSI1, I put a question mark there because there's actually two genes near to each other. We don't actually know which one is which, but it's a polycomb related thing. And then there is a demethylase, Jumondi26, that hadn't been studied before. Okay, and again, more beautiful GWAS results. You're smack in the middle of the genes. There can be no doubt that these are real, but we can also easily confirm it by knocking them out and things like this. Okay, so story thus far. So we found strong geographic patterns in methylation, and now focusing on the TEs, we see that CHH-methylation is influenced by temperature, but there's also major polymorphisms at a small number of loci, right? That affect a lot of this. I didn't show you the figure of this. It only explains the pattern on the map to a relatively small degree, though. There's clearly correlations, but not enough. CHG-methylation looks very similar, but it's a bit more complicated, probably because this trait can be put on, but also maintained in a different way. But why is there so much variation? Well, next piece of the puzzle is transposons. So transposons are also highly variable. So people used to say that you can't study transposons in Arabidopsis because they're all dead, and you should really work on maize because maize is all transposons. So it turns out that there is plenty of transposon variability in Arabidopsis, and they're very much alive. In fact, we find an almost threefold range in estimated. So this is all based on short read sequencing data, which I don't recommend to anyone to try to do. Find where TEs are using short read data. It doesn't work very well, but roughly speaking, we can get some idea how many there are. And you see the map on the left here is just showing transposon density, and there are geographic patterns that are not completely independent of the methylation patterns we saw before. And okay, this one is a complicated figure. Okay. All right, I'll try. Fine. So the rows here. So you take all the accessions, right? And this is what happens when you come in. You get some figures. We ordered all the accessions by how many T's they have in them. And then for each line, we ask, what is the population frequency of the T's that you find in this? What's the distribution of population frequencies? And what you see then is as you go up here, and you can see it on the 3D thing, the ones that have a high load have a lot of really rare transposons, right? So that's what you'd expect if the reason there's a loaded because there's been recent expansion of TEs in those accessions. Okay, and that's indeed what happens. You can see it if you do it by family here, right? So here again, on the plot on the left, you ordered all the accessions by how many T's there are, and then you look across all T's, right? And then for each family, you see how many are in each of its accession. And you see for most of the ones, the highly loaded ones on the right, the same families are up there, right? But there's some families that show a different pattern. And if you put it on the map, you can see that there are local outbreaks of T's in different parts. And the regions that have a lot of T's just have a bunch of coordinated outbreaks by a whole bunch of of transposons, and we have no idea what that is. Okay, so that raises the question, if, can you do a GWAS on transposon activity? So we tried and failed. Vincent Collot's lab did not fail. They were good enough at calling the transposons that they could identify the rare insertions and use them as a phenotype, right? Because they might still be associated with the modifiers. And when they did that, they found one of the hits for transposon activity was exactly the same SNP that we had identified for NRP1 for methylation, right? So you have a POL5 allele that definitely affects methylation and is also associated with transposon activity. So this is a pretty clear connection between what must be going on here. And it gets even better. So on the GWAS plots for CHG I showed you before, there were 5 peaks. They add up, to affect the level of CHG. And so Eriko looked at the transposon mobility, and what she found is that the increased transposon activity is really only found in the accessions that have the alternative allele that decreases CHH-methylation of NRP1, but also has at least 2 of the negative decreasing alleles for CHG, right? And notice that the scale, they go from 0 to 5 on the left. But in this particular combination, you never find all the 5 negative CHG ones together with the CHH reducing NRP1 allele, right? So this suggests that there's some kind of stabilizing selection for not reducing methylation too much. Okay, so story thus far. So we asked about what's all this variation, right? Well, the variation does appear to be related to TE silencing. So at least here, there's a hunch that what we're looking at here is some kind of immune system, right? With lots and lots of variability, somehow trying to keep Ts under control. Why they vary, why there's a geographic pattern, who knows? Okay, but what about CG methylation? So can we understand that also? Because that also shows a geographic pattern, which is sort of weird, given that it seems to be, highly heritable and so on. I mean, there is a lot of papers that basically thinks of it as sort of a SNP, right? People apply a mutation model, there's a mutation rate and so on. So we looked into this a bit more. A student in my lab did a cross between a line from northern Sweden and a line from southern Sweden, differ by at least 10% in gene body methylation. And in this, you do an F2 cross, and what you see, of course, is that for the most part, the methylation status you have is the one you inherited from your parent, right? This is stably inherited. So, these segregate. But not completely. What he realized was that if you actually have enough sequencing coverage, you can see that, and you can already see that in that red curve I had before, that even though sites are at 0, 1, they're not always completely at 0 and 1. And if you look across a whole range of F2 families, you have hundreds of individuals, you have pretty deep sequencing, you can start looking at somatic deviations from the inherited states, right? So on the left, here's just one example. At a given site, here's a site that should be inherited as a 0. But in this case, one individual has actually quite a bit higher methylation. So somehow there's been somatic gain of mutation there, right? On the one on the right, these are things that are inherited as 1, and you can see that quite a few that have sort of lost methylation in some sense, right? And it stands to reason that these differences that we see between, so in this case, between lines, right? They must arise somehow, right? Somatic mutations, well, plants don't really have a germ line, so there must be some probability that this stuff gets passed into the next generation and gets fixed, right? Okay, but the important thing here is that this is a phenotype, right? This is a property of the individual. So now we can do genetics again and look at it. Okay, so we do that. And this is just looking across all the F2 individuals, each row is an F2 individual, and you're just plotting these gain and loss phenotypes. And the first thing you see here is that gains and losses are not the same at all. The losses are found across the whole genome at a fairly uniform rate. Gains not at all, right? The gains are in particular genomic regions. And in fact, you can see that supporting the notion that this contributes to building up the differences. The places where you see change are more likely to be the ones where the two parental lines actually differ to begin with, right? Whereas sites that are zero in both. So this is not independent of this thing at all. Okay, you can also do mapping, and it gets even weirder. All right, so now we do linkage mapping, looking at gains and losses in 500 kb windows across the genome. And what you see is, okay, again, there are cis factors, mostly focused so the gains are on the left, the losses on the right. You see, first of all, there's more gains around the centromeres, so there's some kind of local dependency there. You also see there's LOD score on the top, and you can also see bands going down this way, right? That basically you find transacting modifiers. But the cool thing is the thing on the bottom. If you try to fit a model, so he did this cross in 2 different temperatures. So you can actually fit the model where you try to see, is it explained, by the temperature by itself, by cis, basically the origin of the region. So this is not like regulatory SNPs here, but basically, do you come from this parent or that parent? Transacting factors, trans by temperature factors and temperature alone. So the temperature alone doesn't play a big role, but you can see again, big difference. And let's focus on the losses on the right. Most of the variation we can explain, which is not very much. Again, we have that on the x-axis here. Most of the variation is due to transacting modifiers. There are transacting genes that affect how much loss you have. For the gains, it's really weird, right? And this was completely unexpected because it all fits into a G by E. So there's transacting modifiers that are environmentally sensitive that cause these somatic gains in CHH-methylation. And then you have this weird cis effect, right? That basically there's a tendency for that, or depending on what your mom or dad was, there's a tendency to gain or loss. There's some memory of where you are that causes you to change. So this very much does not look like a SNP, right? If you think about the SNP model, the idea is that basically mutations, mutation rates vary across the genome, right? But still, mutations are supposed to happen independently of each other. They're not supposed to know where they started from. And none of these things are true here, right? There's environmental dependency, that transacting modifiers. We can also show, at least for the gains, that there are close proximity effects, right? If this thing has changed, then this is more likely to change, which is not surprising because what happens here is that there are enzymes that come and do things, right? This is not like random mutation. So, details matter, right? If you're going to model this stuff, you better have the right model. Okay, so wrapping this up then. So yeah, so the plot is thickening here. So I've told you methylation plays a role in silencing transposons. Both methylation and transposon activity vary geographically and are affected by the environment. Methylation and transposons is affected by a whole bunch of major polymorphism in known silencing pathways that basically pretty much look that they must be selectively maintained somehow, right? And that these also affect transposon activity, at least in some cases. Gene body methylation, we still don't know. It sort of looks to me like a very persistent phenotype. It's affected by transacting modifiers. It's affected by the environment, but it probably lasts for quite a long time, right? So we shouldn't be so narrow-minded and just focus on individual generations. But it's still not clear whether it does anything or it's just a side effect of other things. And, the other thing, everything is correlated here, right? You can see that the methylations is correlated with the TE load. When this goes up, the other thing goes down, right? So applying models. So this is where I'm waving my arms quite wildly here, right? There is a sense here that, this is some kind of homeostatic system we're looking at, right? That is trying to keep the genome in order. If you knock it out, bad things happen. Transposons are moving, then non-homologous recombination, meiosis doesn't work, and so on, et cetera, et cetera. There's temperature dependence. I don't know why, but somehow the genome is like trying to cope with a whole bunch of things, right? And if you push this, it's all so these changes are all sort of connected to each other, right? And we don't know. So there's a lot more to figure out here. Is basically we're going to end. So I don't have any answers beyond that. I just want to put my thank you slide to, this is from one of our lab outings. It's Joy Bergelson, if you know her, and lots of people in my lab. So many lab members, collaborators, too many to mention, funders also, and the whole Arabidopsis community, which have been fantastic in this. So thanks.
Hi, I'm Chris, UW-Madison. I had a question related to the temperature dependence, which is, given that the methylation actually physically changes the moiety of the base, could the methylation be doing some kind of steric interference or changing the electronegative properties of the bases such that the binding energy is different? So it's changing the annealing and de-annealing properties of that stretch of DNA, which the CG content kind of has been shown to be responsive to temperature and regulation with respect to annealing and de-annealing. Do you think?
Yeah. It's
part of
yeah. I mean, sure. I don't know. I'm not a biochemist, right? But yeah, we speculated about it. I mean, it could be something simple that you actually, you need to do something. If you imagine that gene body methylation does something, right? Maybe it just have to do with, transcriptional machinery moving at different speeds at different temperatures, right? But then again, right? This could all be driven by, transposons that indirectly methylate this, and the transposons actually responding to viruses, and the viruses depend to aphid density, which actually, we don't know. I mean, it's hard, but yes, absolutely. But yeah, I mean, the more simple one for temperature, right, is that a lot of these silencing pathways are RNA DNA binding, right, and that is absolutely temperature sensitive, right? So yeah,
Thanks.
Hello. Thank you so much for your talk. It's been a little while since I've delved into epigenetics, so hopefully my question isn't too off base. It's another question about the temperature dependency of CG and the other phenotypes you were seeing. So I was wondering if you think that there might be something at play in terms of how the DNA could be coiling and uncoiling itself around the histones, and maybe that rate of that uncoiling might be also correlated to temperature, which might also be a reason why you could be seeing that additional correlation in the methylation of the DNA as it uncoils.
sure. So these short range dependencies I told you about, I didn't go into this, but they are clearly related to nucleosome positioning. So there is basically you see a peak of dependency that's 170 base pairs away, right? So yes, that possibly, but I'm the wrong person to really answer this. I mean, people are really working on figuring this out. Yeah.
Thank you.
So,
hey, that was really, really neat. First off, I want to say thank you for the 1001 Genomes project. If for no other reason than as a teaching resource, it has been incredibly valuable. Really hard to have these kinds of genetic data sets. In light of this morning's talks that we saw, I wonder if you could say anything about the effect size distribution that you observe for these traits in Arabidopsis. And you very clearly made this point that it's so fantastic for GWAS. We know some of the reasons, levels of variation, etc. But are there evolutionary reasons that would be creating a situation where the effect size distribution is so clean in this way that you are seeing these large effects, alleles segregating in almost every phenotype you're looking for? I mean, is this about inbreeding? What's going on here?
yeah, I mean, so I don't know. So, some of you probably know Ed Buckler, right? So we've been talking for a long while about this because in maize, you don't see this. I mean, that's on some level, the simple answer I'm tempted to give is because we pick interesting phenotypes, right? I mean, like I said, I mean,
not like DNA methylation,
No, that are under selection. They're doing, I mean, I think we see such big effects because we tend to look at things where this is basically somehow local adaptation involved here, right? I mean, I played a minor role in a human skin color GWAS, right? And if you look at the skin color GWAS, it looks exactly like this, right? In admixed population, there are major alleles in known pathways shared with mice, you know, that do coat color and stuff like that, and they explain this stuff, right? So that would be the simple one, but that's a little bit too simple, right? Because this is always Ed Buckley's response, because if you look in maze, they also look at things that are often locally adapted, and they don't see big effects. So the answer is I don't know. I mean, but yeah,
Fair.
yeah, thanks.
Wonderful talk. So I wanted to come back to the striking geographic pattern of variation in overall levels of methylation. Since we know that there's local adaptation from the center, or differential local adaptation in the center of Europe to the edges, there's climate gradients, it's so beautiful. We want to believe that there has to be something related to adaptation. And I was wondering, could we also explain this through neutral demographic processes, as we know that Central European populations are a more recently expanded lineage. Can that be related to, I don't know, range, allele surfing of TEs, and then there's like a follow-up of methylation across the genome? Can we, so where are we in this spectrum from this is local adaptation to this was a demographic consequence?
Sure. Well, yeah, I mean, that's a hard one, right? I mean, I think the effects are way too big, right? I mean, if you look at, for example, this T density, there is you know, there are like 3 times more transposons in northern Sweden compared to in Germany, right? Sorry, yes. So yeah, so okay, so about, it's very hard to know, right? You know, whether these are neutral things. If the differences are very, very big, right? So and if you at least if you apply this kind of QFST or like various kind of permutation methods, it's pretty easy to convince yourself that, yeah, this doesn't look neutral. I tend to be very skeptical about all this stuff, right? So, you know, but I would bet a lot of money that it is not, right? But I can't really say more than that. I mean, if you read our papers, we usually try to do things like, you know, permutation tests, right? You can, for example, see that I showed you that there was an anti-correlation between this NRP1 allele and these things. I mean, if you simulate that, you draw random snips from the genome with exactly the same frequency, you know, you never see that pattern, right? So, right, for what it's worth, that's one way of doing it. But of course, it's a post-hoc test. So, you know, right?
Is there any relationship following up on that of the deleterious loads in populations that are recently colonizing with some type of levels of methylation CHH or
no, I don't
activity?
think so. I mean, we've also done
Because that
studies.
would be a way to kind of.
We've done reciprocal transplants and mapped fitness effect in genes, and no, I mean the lines up in northern Sweden are for sure. You know, they live in a more fragmented environment. They're much more extensive LD, longer haplotypes, and so on. But they're definitely locally adapted to their environment, right? So, you know, probably there are also more deleterious mutations, right? But you know, Arabidopsis has such a weird. I mean, it's a matter of scale too, right? I mean, on a very local scale, you can of course find deleterious mutations because you can get, you know, it's a sulfur, you can find very strong founder effects locally, right? But, you know, if you look at synonymous, non-synonymous differences, there's not the big change geographically at all.
Thanks.
Hi, I'm Mariano from Avalo. Thank you for the talk. I just had a question about, I'm also kind of interested in what methylation is like doing. And there's a lot of really beautiful GWAS work here, but I wonder if there's anything to be learned from sort of turning it around and using like a genomic prediction model and then asking how the deviance in predictions from the genomic data versus some of the epigenomic data might explain a little bit when the environment is when methylation has a relationship to the environment that is sort of independent partially from the transposable elements and from the genetic variation.
Let me see if I understand that. So you're asking if you can predict where they're living based on.
Yeah, just that I think like sometimes people have used like when genomic predictions, for example, are doing better or
Right,
doing worse, and there's like information, the
Sure.
subtraction between the.
So I can say one thing first, that polygenic prediction doesn't work at all for our purposes. There is far too much linkage to equilibrium between things. There are way too many big effects. It doesn't work. But of course you can predict things given that we have such big effects. I mean, you can plug in, these 5 major alleles and things like that and try to do things, right? And I mean, yeah, they're correlated with all kinds of things. It's just, I haven't done too much with it because we're trying to, I'd rather try to do some field experiments to get direct evidence, right? Because everything ends to be correlated with everything. And yeah, but the data are available. So if anyone wants to, play with these things, it's,
Cool, thank you.
Let's thank Magnus again.